when we talk about depression, of course, it means a lot of, you know, when you want to describe depression, perhaps you find yourself in relation to many different forms, terms, feelings, and concepts, like low energy, poor concentration, depressed mood, disturbed sleep, poor appetite, although sometimes it is higher appetite for some people, low self-esteem, hopelessness, weight change, not necessarily always weight loss, but also gain, feelings of guilt, thoughts of death, anhedonia, that you are not able to get any pleasure from anything, and agitation. And actually these different symptoms can also be sometimes described according to maybe the subtypes of depression. Next slide, please. So first we will talk about the definition, prevalence, and the burden of depression. And then we are going to talk about the neurobiology of depression, in which we are going to have overview, especially with emphasis on genetics and brain regions associated with depression. Then we are going to talk about the theories of depression. But long story short, I want to say first, despite all these things, and despite all the things that I'm going to talk about today, we really don't know the neurobiology of depression. Okay, that's the first thing. I think so far, based on my classes, you understand that we are dealing with mental phenomenon and its disorders, which is extremely complex. And you know already the difficulties associated with that. So you would expect that actually we don't know much about it. That there are many hypotheses, there are many ideas, there are a lot of data. Some of them are not consistent, some of them are not validated, some of them are not replicated. Still, we have some, you know, window that is being opened, and maybe there will be some more progress in understanding of depression eventually. Next slide, please. Previous slide, sorry. So if we have, so the first two topics are going to be covered today, and if we have more time, we are going to talk about the theories. If not, next class, we are going to talk about the theories of depression. So, okay, so the definition. So, you know, you may feel depressed. You may find somebody depressed, but in order to diagnose somebody clinically, there are some criteria. And those criteria, as we already discussed, are determined by Diagnostics and Statistical Manual of Mental Disorder of American Psychiatric Association, and based on some regular periods, it is updated. And now, actually, we have depressive disorders, and you see the list of depressive disorders here. Let's first get aware of disruptive mood dysregulation disorder, major depressive disorder, persistent depressive disorder, premenstrual dysphoric disorder, substance or medication-induced depressive disorder, depressive disorder due to another medical condition, other specified depressive disorder, unspecified depressive disorder. So the major depressive disorder is actually the depression that I'm going to talk about during this class. But you already realize that actually there are many subtypes, especially, for example, think about, okay, this is clear. And this is not to be confused, even in the clinical studies. It's very clear that if somebody would like to understand the pathophysiology of depression, these group of patients, which is due to the cyclic menstrual dysregulations, is not supposed to be considered maybe under the you know, title of depressive disorders, especially if you would like to understand the pathophysiology of depression. But think about this and this. So, like, you know, this is actually just differentiated from the major depressive disorder based on some symptoms and maybe the severity of the symptoms. And that is very hard to actually sometimes clearly, you know, set the borders in between different diseases. And also here, when you talk about these diseases, age is very important Sex is very important. Not only the age, but developmental stage is very important. For example, how one form of disease is appearing during the period of adolescence might be very different when you are an adult, when you are old. So how the symptoms and biological also indications or relations or associations of this particular clinical case is reflected is different. So you have to be aware of this. So most of the time when I talk about depression, unless something else is indicated, I mean major depressive disorder. And sometimes in some researchers, which I'm going to present today, then I will talk about mostly, for example, treatment-resistant depression. So some people have also depression that is not responsive to any treatment, for example. And those people sometimes are treated with deep brain stimulation. And we also get answers from that. So most of the time, even those treatments and the data from those treatments in this class are used not to explain you what kind of treatments exist, because as I said, I'm not a clinician, but we use those data in order to understand the pathophysiology and mechanism of depression. So if certain people, as a result of specific stimulation of certain brain regions, relieve their depression, that is scientifically also important to understand the mechanism of depression. So there must be something related to depression there. Okay, so please keep the context in this, context. Let's just have a look at the criteria for the depression. So, for example, this is according to Dsm criteria. Here you have the 9 different criteria, and actually a person should have either A or A1 or A2 together with other criteria, 4 of them, in a certain period of time, and some with the details of other also conditions. But let me say, so the person should have depressed mood, indicated by subjective report or observation by others, or loss of interest or pleasure in almost all activities. So the person should have either this or this, plus 4 of these, such as sleep disturbance, so not necessarily insomnia, which is not to be able to sleep, but also hypersomnia, so sleeping more, then psychomotor changes such as agitation, severe enough to be observable by others, tiredness, low energy, decreased efficiency with which routine tasks are completed, a sense of worthlessness or excessive, inappropriate or delusional guilt, impaired ability to think, decreased concentration, so you're not able to concentrate, recurrent thoughts of death. So either this or this plus 4 of any of these, okay, at least for 2 weeks. And that is not happen. And also, the severity of these symptoms should be severe enough that the person's social and professional life is being interfered with this kind of indications. So I think now everybody is clear more or less what is the criteria for depression and what is the prevalence of depression, how actually commonly, it's a common mental disorder, and globally more than 264 million of people of all ages, so age is very critical. You already know the brain development, how it has these landmarks. Of all ages suffer from depression. More women are affected by depression than men. Depression can lead to suicide, okay? There are psychological and pharmacological treatments for moderate and severe depression, but we are going to discuss that there are problems associated with this statement. So it is not really so straightforward like this. But there are, yes, there are some interventions that helps. now the burden of depression. So the burden of depression can be explained in terms of years with disability, especially this one. The burden of living with a disease or disability is measured by the years lost due to the disability component. And for that, for 2017, there was this statistics which is called the Global Burden of Diseases, Injuries and Risk Factors study for 2017, GBD study, which was actually assessing the different diseases, especially, I think, how many diseases? 354 causes of disabilities, okay? And then they ranked which kind of states, diseases, and conditions were actually more, causing more burden. And what was the rank of depression among all these? So, back pain, headache disorders, and the depression. So depression was number 3 across the 354 causes of disease burdens globally. And that has become, in 2019, number 2. Okay, so that is like after even 2 years, it became number 2 in terms of years lived with disability. So it has a huge burden in terms of sustainability, in terms of life quality of individual and society and everything. It's a serious disease. So the problem is it kills you without killing you. Slowly, slowly, slowly. Every day a bit of. And that is a huge impact, the huge burden, and as you can see that here. So that's a serious problem. So, despite this, we still do not know much about the neurobiology of depression. Long story short, the reasons for that is because of the lack of the biological markers that could explain, also guide biological research. We need to identify biomarkers by biological research, but in order to identify biomarkers, we need some other biomarkers. So it's like a circle. And the heritability of depression, okay, it's heritable, but for example, compared to other mental disorders such as schizophrenia, it is relatively lower in terms of heritability. It has relatively lower heritability, and that makes a little harder to find the genetic link of such a complex disease. And of course, the complexity of the other factors that are associated with depression. But that is already we know for most of the mental disorders, as you remember. The complexity of the genetic factors, the complexity of the environmental factors, and the complexity of their interactions. That is amazingly complex. That is the problem, and that is not exception for depression. So, okay, what is the heritability, of depression? Heritability is defined as amount of observable variation in a population that is attributable to individual genetic differences. That is the definition of heritability. And they have found that the heritability of depression is about 38%. Okay, so if there is a heritability, if there is a genetic component for the etiology of depression, so is there a depression gene? What do you think? Yes, that's a good answer. Exactly. So everyone in this class knows that that's the wrong question. That was a question to actually make you mistaken by purpose, and you gave exactly the right answer. We cannot just say, after taking so many hours of biopsychology, we all know that we don't deal with depression gene. No. This is a common disorder, this is a complex disorder, and that is probably, related to probably thousands of different genetic factors and their complex interaction, plus their interaction with the environment. But many, many times, people were actually challenged by this question. If you go maybe 20, 30 years before, you would find that people were asking this question. And you may find some, headlines, oh, the depression gene was found. This is a mistake of popular media, because they oversimplify the things sometimes, but that is even so simple that it is not true anymore. So there is no such a depression gene, but what is it then? So the problem is that was this phenomenon called the missing heritability, because on one hand, we had this idea that there is almost like 40% heritability of depression. On the other hand, many studies have been conducted, at least until now. Because now I will, in a few slides later, I'm going to give you some good news. But actually, until recently, despite all progress in the genomic technologies, we were not able to find actually the association of genes with depression. Many studies were done and many efforts and many findings were produced, but they were not replicated by other findings. They were not validated by other findings. And that was like huge data with a lot of false positives. Now, let's have a look at what are those methodologies that people were trying to find out the genetics of depression, or genes that are associated with depression. Traditionally, there are three kinds of studies. One of them are the linkage study, the other is the candidate gene association study, and the third one is the genome-wide association study. In the family linkage study, they follow the co-segregation of disease in a family together with the genetic markers, and then they make a kind of biological hypothesis based on that familial co-segregation. In the candidate gene association study, what they do, here you have to think the biological hypothesis in the center, and you have to look at the direction of the arrows. So in this one and this one, you start with a kind of, no hypothesis. But here, especially in the candidate association studies, you have to start with an initial hypothesis. And that hypothesis would be, okay, some previous data show that depression might be related to serotonergic system genes, or one gene that is actually found in the serotonergic system, because the drugs that are interfering with the serotonergic system works well on depressive patients. So that's like an hypothesis, how they move. And based on that, what they do, they take the depressive patients, they take the healthy people, and then they isolate their DNA, and they analyze the gene, the candidate gene, for example, serotonin transporter gene. And they find out what kind of variations exist in the cases and in the controls in terms of that one candidate gene. And actually, this candidate gene is having not only one variation, it has multiple variations, thousands of variations, depending on the location. But they choose just one or few variations in different experiments, because at least until this moment, we didn't have such a technology to check all variations at once. So then they compare if a specific variation of that gene is more abundant in specific diseased cases of people compared to the healthy controls. Okay? So that's a typical candidate gene association study. All right? So there were many studies that have been done by this way, but they were not really producing good results. And most of those results give so many different genes and associations, but eventually none of them were validated. So it was a lot of effort without any fruitful result. We will discuss why, but now I would like to explain you genome-wide association study. In the genome-wide association study, you don't have any hypothesis. You don't say, oh, this gene or that gene. You just have nothing. And what do? You get patient group like this. But there's a big difference between the cases here and here. Here, you deal with 10,000 people, for example. Here, you deal with sometimes 100 people. Sometimes even in the earlier studies, less. But here, you deal with 10,000 people, and those people are, very carefully and systematically selected based on age and sex. For example, they say, okay, I'm gonna work, we are already dealing with a very heterogeneous disease, so I'm gonna work only with the females. I'm gonna work only those with a certain age. I'm gonna work only those who have, for example, treatment-resistant depression, and I'm gonna have 10,000 of them. Okay, why we need so much big data? Because it's so heterogeneous that in order to find out one simple relation, you need to really look at the big picture. And here, what is really big is not only the sample size. Another big thing is that you don't check only one gene. How many genes do we have in the human genome? Approximately 20,000. And how many genetic variations we have? Think about that. It is much more, much more. Millions of genetic variations are going to be scanned in thousands of, 10,000s of patients in the cases and controls comparison. So that requires a huge computational and statistical power also. So here you see that we have methodological challenges to do that, but thank God we have both technical, statistical, computational effort or progress to do such experiments, and that was done. So that's why this is called Genome-Wide association study. These kind of studies, because of the progress that we have in the last decade, caused the identification of many important genetic components of several complex disorders. But depression in the beginning was not among them. But later, a better design of the genome-wide association studies led to the identification of actually certain genetic factors that are thought to be associated with depression. But before that, I want you to be aware of the big data concept, okay? Big data is a field with the systematic extraction and analysis of data sets that are too complex and large for traditional data processing systems. So big data can be applied in different fields, but big data are being applied in psychiatric research as well. Okay, so psychiatric research was using the big data applications, storage, analysis, and retrieval of the medical and scientific and biological data sets. But that requires one important thing. Then the psychiatric circles have to work in consortiums. So one consortium, for example, is the Psychiatric Genomic Consortium. Psychiatric Genomics Consortium, that means more than 800 scientists from 150 institutions in more than 40 countries. And those are linked to the hospitals with all these patients. Then they have to collect all the biological data that are associated with these patients. When I talk about the biological data, what do you understand? For example, they take their blood, they analyze, they have the all blood material and purified DNA, and they have the access to analyze all variation, genetic variation individually for each patient. But also they have other data like age, like, basic demographic and personal properties of the individual's lifestyle. They have other biological data such as brain scans. That is also a very important kind of data set for these kind of consortiums. So they scan the brains one by one, but you need a lot because it's very complex. And in order to unveil that complexity, you need to work with the big data to find out some meaningful, maybe the most prominent or most associated genetic variations. So what you have to realize that the big data is also used and utilized for psychiatric research that require extremely well systematic knowledge and data gathering and working in consortiums, and that is very common. Today, it is one of the example is this, but there is also other consortiums In all different mental disorders, now they work under like consortiums actually. It's very common in biological research today to work as consortiums. So that missing heritability that somehow was found. Why do we call until now as missing heritability? Because twin studies show that there is a genetic component that is heritable, but somehow what are those genetic components? We were not able to find out. Despite all effort, we were not able to find out. And that's why there was this period of time that heritability was described, that there seems to be a heritability, but we were not able to identify it. Either heritability estimations were wrong, that there is no heritability in depression, or we are not able to find out because we need better techniques. And yes, as a result of genome-wide association studies, even the first lines of the genome-wide association studies were not able to catch those components, but a careful, better design of these kind of experiments eventually led to, at least there are others, but for this class I just selected you to 2 examples. Here you see this number, RS 1241580. This is actually genomic location of a specific genetic marker or single nucleotide polymorphism that is, for example, found in the chromosome 10. They found that as a result of this enormous data processing, they end up some variations that are statistically significant enough that to be considered as associated with depression. Another one is the second one, and this is, for example, this variation is located in a gene, near the gene. So not everything in the human genome is considered as gene. Certain things are not encoding as genes. So these are called non-coding regions. And one of them is located near the gene called SIRT1, and the other one is located in the intron. Well, maybe you don't know what intron and exon, because you know mammalian genes have specific regions, intron and exon, and introns are normally the regions that are not directly coding the proteins. When the messenger RNA is transcribed from the DNA, the introns are removed. So basically, you would just think that the introns are not directly important for the protein and its function. And so far, actually, we were thinking that the non-coding regions, both of the cases here, we didn't think that they were so important in human genome. But it looks like they have some importance that need to be discovered, as it is also indicated by this case. Why anything that is directly on the gene coding regions were not actually discovered, but all these regulatory regions were discovered as associated with depression? That's an interesting question to ask. Let me show the location. So this is the location, this is the chromosome 10. Both of these genetic variations that are thought to be associated with depression are found in chromosome 10. This is, do you see this red color here? Maybe yes, some, maybe no, but it's exactly here. This is the chromosome 10 and the location of this variation. Okay, and this is again in the chromosome 10 and location of the second variation. It's here, somewhere here. And I took that from the gene card. If you click on these links on the lecture slides, when I upload it to the Canvas, you can also find and explore all the information that you need to find about these genes. So this is one of the latest findings about genetics of depression that are thought to be associated with the depressive disorders. So this is like, and forget about the previous ones that are found by the candidate gene association studies because they were not replicated. So my next question is, what are the brain regions associated with depression? So first we approach the problem in terms of finding association of genetic variation by comparing the diseased people and healthy people. And for that, we had two approaches. The first one was candidate gene approach, that we already know which gene should be associated. It was a preliminary hypothesis. And then we checked that gene variation with a design of a case control study. But that didn't lead any fruitful conclusion. And after that, the genetics have been studied by Genome-Wide association studies. And based on that, as I already summarized, we have found some fruitful results with high statistical correlation. And that were described, that they were found in the chromosome 10. Is that all? No. There are, as I said, it is not only these two variations. There must be many different variations that are associated with depression. And but for the moment, that is what we have, and that's a good progress. Now, of course, not all the studies that are trying to understand the etiology of depression are only checking the genetics. There are many ways to do biological research to understand the pathophysiology of depression. So another way is actually to check the brain regions that are associated with people. So what do? You just check the brain scans of the people and compare it with the healthy people. And the brain scans can be checked in terms of structural and activity-related measures. Structural means, for example, what is the volume of a specific brain area in normal people compared to the depressed people, for example. Functional brain scans are also possible, in which you actually find out what is the activity level of people who are depressed compared to the normal people. And that activity level can be measured in terms of a specific stimulation. For example, you can show an emotional stimulus. What's an emotional stimulus? For example, you can show a traumatic movie, traumatic photo. Okay, and then you measure the brain activity pattern of individuals who are depressed and who are normal. That is the way you can compare with the functional changes. Let's have a look at what they have found. Yes, they have found consistently, so these are the consistent findings, that in terms of the volumes, that there is a reduction in terms of the gray matter volumes in the brain area, which are prefrontal cortex, hippocampus, amygdala, and cingulate cortex. So this is the sagittal section of the brain, as you remember, and this is the prefrontal cortical area. This is the cingulate cortex here. Okay, and this is the amygdala. Okay, this is the hippocampus. As you know, amygdala and hippocampus are the brain regions, including cingulate, that are the limbic system. Especially the cingulate was specifically associated with depression in terms of its anterior part, which is the anterior cingulate cortex. That was actually, the activity level was different compared to normal people. But here we only explain them in terms of the volumes. So the tissue, there was a reduction in the gray matter, the nuclei of the cell bodies of the neurons. So neuron numbers were decreased in these brain areas for people. Now, my question is, let's say you have somebody in your family who is depressed. Do you think that person would exactly have all these things? No. This is a cumulative result of also probably, you know, very specifically selected a systematic subgroup of depressive people. All right, so as you already know that it is so heterogeneous, there are so different factors, so many different factors. So you cannot really say that every individual who is depressed will 100% have all these things at once. So, but yes, based on a very severe and systematic cumulative results of the comparison of the brain regions in terms of gray matter volumes, we really know that hippocampus, amygdala, and prefrontal cortex have been studied very well, and there were, like, you know, gray matter abnormalities in these regions. What about the function? So when you measure actually the gray matter, it is like, you know, what is the volume of specific brain area? But what about the activity of those brain areas? How are they correlated with depression? So what they found again is that, again, this is a cumulative result, and it's not necessarily the case but for every single individual. But in general, what they found that, you know, the cerebral cortex of the depressed people are less active. But there is especially one region significantly less active across the whole cerebral cortex, and that is the prefrontal cortex, as it is shown with the blue color here. So the prefrontal cortical area is slow. And one area associated with emotions and motivation and reward, which is the limbic system, is more active, as it is shown by the red color. So you have somehow a speed up and slow down of different brain regions at the same time. So the prefrontal cortical area is less active. In the meanwhile, the subcortical limbic areas are more active at the same time. And that is interesting because normally the prefrontal cortical area is thought to be, like, you know, involved in many important functions, especially cognitive control. Cognitive control. So, you know, actually, so it's like, you know, the bus here. It has the main thing. And is it because this gets out of control? This is like, because this is too active. Here, actually, I wanted to show you the regions that are slowing down and speeding up during depression as a result of this previous activity study. Here you see the prefrontal cortex, but we put it like a transparent structure because underneath you have the limbic structures. Of course, hypothalamus here, but of course it is not the ones that I want to show you, but look at the cingulate cortex here on the right and on the left, and then look at the hippocampus, fornix, septum, look at the amygdala, and look how they are connected with each other. I mean, maybe it is just one of them is overactive and the others not, but they are all affected, and but basically we know that this area is overactive and the frontal cortical area is less active. That is measured by the functional brain imaging studies. Now, I want to give an idea that you may consider that actually even the previous slide was too simple. What is brain activity? And it's like actually has to be studied in many different domains. And that is an example for you to understand how actually in different levels of complexity this situation can be analyzed. Let's take hippocampus. What happens to the hippocampus when people are depressed? Again, in this certain group of, you know, very systematically selected specific group of people, especially with severe depression. So, impairment of Ltp in the Ca3 region. We already covered what Ltp is, the mechanism of synaptic plasticity dependent on activity. Facilitation of Ltd in Ca1 region. So Ltd is another form of plasticity observed in the hippocampal cells, which is long-term depression, that the strength of the synapses are decreased. And somehow that mechanism is facilitated. And then downregulation of synaptic proteins and growth factors. So what does it mean? That they, you know, analyze the content of the synaptic proteins compared to the healthy controls, and they found that there is a decrease in the number of synaptic proteins and the growth factors. Growth factors interesting because growth factors is related for the formation of new neurons, and there's this downregulation there. Is it because of the stress that the person is exposed to, or is it because actually the other way around, which is the cause, which is the effect, is not easy to understand. Volumetric changes, as I already described you before, but it is just actually an explanation that how the changes can be studied in very different levels. And I was just showing you the volume of hippocampus was decreased. But you see in the cellular functional level, synaptic level, in the other levels, there are all differences, and they all have to be mapped together to make a meaningful, useful relationship. Neurogenesis, the formation of new neurons. What happens is that hindered by high levels of glucocorticoids and enhanced by adrenaloctomy, additive effects in mice while reduced in humans, additive function in the circuitry. So these studies also done in both animals and humans. And apoptosis. So apoptosis is actually the opposite of neurogenesis. Neurogenesis is the formation of new neurons. Apoptosis is that of the neurons. Apoptosis is also natural. Cells have a cycle, and after that they die. But the depression promotes apoptosis in the hippocampus. That means that there is an abnormal process of cell death. And the effects caused by chronic depression last longer than those of acute depression, for example. So the chronic is the long term. Acute one is just which you have an episode. What about the prefrontal cortex? So what kind of changes were actually observed in the prefrontal cortex? Again, you can actually see activity. I was talking about the activity patterns already, but actually you see there are so many things that are actually considered, that should be considered when we think. So I was just simplifying it to show you, okay, prefrontal cortex is slower, and the limbic structures are acting in a faster way. But actually, you see, the prefrontal cortex have to be examined in terms of the composition of receptors in different brain areas. It has to be examined in terms of downregulation or upregulation of proteins and their types. It has to be studied in terms of, for example, different neurotransmitters and their synthesis, their storage, their activities. It has to be also addressed in terms of many other parameters. These are just example slides for you to show that, just to simply say that, oh, we have found 2 genes that are supposed to be associated with depression as a result of genome-wide association studies. And also we know that, there is atrophy in selected brain regions. And then also we know that brain regions that are found in the prefrontal cortex are less active, and subcortical limbic structures are more active during depression. So it's not so simple. But I don't expect you to know all these things, but I just want you to be aware of that, the complexity again. It's all about being very complex. This is amygdala as an example again. For example, when you have in the amygdala, the increased expression of BDNF, that's a specific protein that is involved in the triggering of formation of new neurons, neurogenesis, okay? And disrupted glutamate signaling at the Nmda receptor. I think you remember the Nmda receptors when we talk about the activity-dependent synaptic plasticity. And that was in the hippocampus. And you have the same Nmda receptors, but this time they are in the amygdala. And their somehow signaling is disrupted, that they have found. Of course, volumetric changes. Larger gray matter volume in the bilateral amygdala. So there was like, in some studies you may also find that some results are not consistent. Okay, because as I said, it is very heterogeneous and you may end up different results. So still we need to clarify these results. And the functional connectivity. So functional connectivity is, for example, how the activity of the one brain area is connected and controlling and interacting with the other brain area. So in that case, decreased bilateral amygdala, right insular cortex connectivity have been detected. In the left amygdala, the functional connectivity decreased in the positive network and increased in negative network. And in amygdala-associated brain circuits, there may be change with depression severity, they have found. So the more severe the depression, there is more actually disruption in the amygdala-related circuitry. So this amygdala looks like, you know, I spend so much time about amygdala, and when sometimes I get out of control, when I'm very angry, very sad, or whatever, and sometimes I'm not able to stop it, then I just think and dream my amygdala, and I feel like I take it with my hand, you know, this little structure that is found just in the anterior part of the hippocampus, and I start to think and dream as if I'm like, you know, when you do massage to your hand or back, just keep your amygdala with the hand and say that, oh, you are too active, my dear. That's not okay. You are too active, and you are just repeating the same pattern all the time. You are out of control. You are, you know, you are actually invading the whole brain. So sometimes you think that your ideas, your obsessions, your thoughts, emotions are invading you. You're not able to think of anything else. You're not able to do anything else. It's probably all these circuitry is somehow overactive. And yeah, the point is the real freedom comes when you learn how to control it. But if you are not able to control it, maybe there is a pathology there. So there is something when the pathology starts, when it is normal, and how we can actually even make it better. There are borders for that. In this slide, we see actually it's a summary slide about the depression. That's what I have covered so far. So in addition, I also mention here the deep brain stimulation studies as well. And altogether, it's a quite well summarized slides of what I have talked in terms of brain regions associated with depression. So here you can see that in this area near to anterior cingulate cortex, okay, they have, and also in the area called nucleus accumbens, when they stimulated those areas with electrical stimulation, with a surgery, they insert the stimulating implants, and they do it only with patients who are treatment resistant. So they are not treated by any other thing. No psychotherapy is working for them, no pharmacotherapy is working for them, and they are in a very bad situation. Let's say every month is like committing suicide, for example. And then they try this kind of methodology, and that helps. That helps for the relief of the symptoms of depression. And that's why we were thinking that those brain areas may be also important to consider to study the neurobiology of depression. Another important thing is actually the hypothalamus. You know, hypothalamus is associated with the stress response of the body. So we will talk about this actually later in more detail, but when the circuitry associated with stress response that is triggered by the synthesis of hormones released from hypothalamus, there is a feedback loop, and that loop sometimes overactive, is not functional very well. The way why that feedback loop is not functional very well is also complex. It could happen that during development, that loop is not developed properly, and it's already oversensitive. It's not working very well. Okay, the stress response mechanism, that is those circuits associated with the stress response during development, is not properly structured or connected because of the severe environmental factors. For example, think about war. people have wars. And then, there are little children, and then they have to be, there are trauma that the parents run away, they go somewhere else, children are separated from their parents. That's a serious stress. And that could make, and during that happens, that trauma happens during development. When you are adult, the way how your brain would respond to the same situation would be different than when you are a developing brain. Developing brain is highly sensitive, more sensitive than the adult brain, because it is being shaped according to inputs. So that's why too much stress is going to distort the development, the natural development of those all systems, including the stress response-related systems. And once that system is not properly adjusted, then in the adulthood you are oversensitive. And then extra stress during the adulthood will just end up with the breakdown, for example. And here is an example for the actually studies associated with amygdala. Here you show this kind of face, like this, fearful face. So the way, and that is here, this color shows how actually individuals respond. Okay, the people who are depressed and their amygdala is actually giving more signal to the emotional stimuli compared to healthy people, for example. Here actually I would like to show you one example from the previous slide in more detail, when we have different brain stimulation treatments for the people who had been diagnosed with the treatment-resistant depression. And these studies are still in the clinical phase, so they are not really actively used for the treatment. So they are trying on certain groups of patients under certain clinical tests. And they have found that not only, in the cingulate white matter, but also in the nucleus accumbens, these two areas were already shown in the previous slides, but there are also two more regions that are shown in this figure. You don't need to remember these names, but actually these four, 1, 2, 3, and 4, these four areas, these four specific areas, when they are stimulated with the deep brain stimulation, they cause the relief of the patients who had treatment-resistant depression. But again, this is like a very specific group of patients that are not responding to pharmaceutical or psychotherapy-mediated treatments of depression. So it's not necessarily to be for other people. And plus, it has some other problems, ethical problems, side effects. That's why these treatments are still in the clinical research phase. The next is actually, so far, we were discussing about the genetics of depression. We know that it is very complex. Then we discussed about the actually brain regions that are associated with depression. We understand that it is actually extremely complex. It is not like this area, right? And it has very different levels of complexity. So if there's an association, that association has to be studied in different domains, like in the molecular level, in the cellular level, in the synaptic interaction level, in the brain activity level, in the network level. And you already seen that example. And now, actually, I would like to give you an overview of the different theories of depression. How many minutes do we have? Half an hour? Okay. I think half an hour wouldn't be enough. I will just give a brief summary today about that. Then we will continue with this in the other class. But basically, we have different theories of depression, and that is natural, right? Because we don't know the basis, neurobiological basis of depression. That's why all these are different hypotheses that are having some background, some biological background, that are thought to be strong enough to hypothesize. But normally, if we know it, we would only have one hypothesis. So we don't know it.
Are they contradicts?
Not not always, I wouldn't say, it could be, but obviously it cannot, they are not also contributing to each other, that they are not, that they are separated. So one of them is very oldest one, actually, the Monoamine Hypothesis. I think it is very easy for you to understand because all the accidentally discovered drugs of depression were acting in the monoaminergic system. And then we thought, oh, maybe there is this deficiency in the monoaminergic system. And why do you think this theory was not enough to understand the depression? Because one reason is very obvious. If this is a real reason, then why everybody who is treated with the proper antidepressant is not becoming okay immediately? First of all, why we cannot get the direct response immediately? Why it takes 3 weeks? The other reason is that why it doesn't work in every patient. So it doesn't work straightforwardly in every patient. We discussed this a lot, you know. We discussed that many times. And it is like an error and trial phase when you want to prescribe the drug for the patients. So because of that, okay, we have this hypothesis, but it stays somewhere. And the other one is the HPA, hypothalamus pituitary adrenal system hypothesis. And somehow this is something I already briefly mentioned in the previous slide, where you have a kind of, this HPA system, which is very vulnerable during development, and if you also have genetic susceptibility as well, and during the development, if it is also, if the individual is exposed to extreme stress, then this system is already not functioning very well. You are okay, but it's not functioning very well when you are adult. And then you are under the risk of the depression. That is the second theory. The third theory or hypothesis is the neurogenesis hypothesis. It is because they found that many antidepressants, when the patient is relieved from the depression, they have found that, especially in the animal studies, the antidepressants are actually creating this effect because of the rate of neurogenesis is being increased. So they thought that, and also they found that there is an association between the decreased rate of neurogenesis in individuals who are depressed, as I already mentioned many times. And because of that, there was this neurogenesis hypothesis was proposed. And then the GABAergic deficit hypothesis. Like the GABA is one of the major inhibitory neurotransmitter in the central nervous system, and there were some findings which we will discuss that could be associated with depression. And the other one is the microbiota and inflammasome hypothesis. That is very popular. Maybe you have heard of it. I am sure you heard of it. You know the prebiotic bacteria? Exactly, the gut-brain connection, it's so popular, it's a hot topic now. So because we have in the gut a lot of microbiota there, and that is actually unique to every person. Did you know that? So that is also very unique to every person, it looks like. For example, when you have antibiotics, if you get sick and you got antibiotics, and that antibiotic is also killing all the microbiota that you have. And then you may be prescribed to get some prebiotics to recover your microbiota. You know that, right? And actually, the point is every individual is not necessarily having originally the same content of microbial cells in their gut. And to which extent it is really something that you can recover is also a question. So you have to be really careful about what you are about antibiotics, because obviously there is a connection between them, between the gut and the brain that we will talk, but obviously they found some systematic results as a result of the microbial content of the gut of the people who are depressed compared to the normal people. And that makes me consider the fact that actually diet is extremely important. Extremely important. And somehow I never hear anyone, sometimes I have people in my surrounding who are officially diagnosed with depression, but I never hear that their doctors are telling them that, oh, you are depressed, now you have to really take care of what you eat. But if you have a heart disease, or if you have some, problems in your veins or liver, oh, you have a special diet, you have to do sports, you have to do everything. You really have to take care of your lifestyle, isn't it? But when you have a brain disease, nobody is really caring about your diet and lifestyle. And I think this is a very important and underestimated topic.
No, they care about lifestyle, sports.
Yes, but not as much as, I don't know, I think not as much as, I know that, for example, my grandmother, when she had heart attack, I know that how this, in addition to the prescriptions, drugs, how her lifestyle is so much carefully organized. So doctor told her every day, there is no excuse. Every day you have to work 40 minutes. You have to eat this, you cannot eat, I don't know, that, and that was like very clear. Do you think any patient who is depressed getting such kind of lifestyle, they just say that do some sports, whatever, they don't give much detail as if the brain is less sensitive than heart? That is the point. So just do some sports, try to walk. No, it's not like that. It's a serious thing. And that is not being realized very well, but it will be realized. This is an organ, this is a biological system, and it's even more complex than heart. So that means that we even have to take care of more, even at least like we care when we have a heart disease, we have to care of our diet when we have depression. It's extremely important for students as well, even for normal people. If you want to really you have cognitive function every day, you are learning, and you really have to have a proper diet. But I think in Russia, there is a good diet. I observe the diets of different societies. To be honest, I have been to different countries and I observed that. So I think it looks okay. It's not so bad. The traditional diet looks healthy somehow. Here, for example, it's very healthy. So we will talk about these 1, 2, 3, 4, 5 of them. For the moment, I want to stop here.